Hello, everyone, and welcome to BrainMap. As you all know, this seminar series is co-sponsored by the P41 funded Center for Mesoscale Imaging housed at Martinos. And I will not introduce the speaker today. I will hand it over to our own Susie Huang to do that, to give us the honor. And I hope you all enjoy this. And please let us know how things are going, and we will be in contact. And if you have questions, please feel free to ask them in the middle. Put it in the Q&A box for people on the Zoom, and Mladen has told us that this is going to be an interactive talk if you want to. Okay, I'll hand it over to Dr. Horn. Thank you.
Hi, everyone. Thanks so much for joining BrainMap today. It's really a great pleasure today to introduce Mladen Barbic, who is now Director of Research Engineering at Tech for Health at NYU. I first got to know Mladen when I was a grad student at UCLA, and Mladen was faculty, I think, at the time at Cal State, Long Beach. Yeah, so Mladen did his undergrad and PhD at UCSB in physics, and then a postdoc in applied physics at Caltech. After that, he obtained his faculty position at Cal State Long Beach, and was there for a few years, got tenure, and then actually made a complete switch and moved over to Janelia Farm, where he worked in the Division of Applied Physics and Instrumentation for 11 years. So he's now been at NYU for the last 4 years, and he's going to talk to us today about his work on magnetocaloric materials as switchable MRI cell tracking microlabels. Thanks, Mladen.
Thank you so much. Thank you so much, Susie. It's really an honor to be here. Thank you for the invitation, and thank you for coming. And hello, everybody on Zoom. So yeah, it's fun to see. I met Susie when she was a grad student at UCLA long ago, and now my son started his undergraduate studies at UCLA. So it kind of shows you time flies. So today I'll talk to you about really a passion project of mine for the last 8 years. And I hope you find these materials interesting for MRI. I sort of fell in love with these materials. I thought their features were quite dramatic and would turn out to be dramatic for MRI. And that turned out to be the case. And so I hope you enjoy it. I do want to mention that this is the BrainMap seminar, and you probably see a lot of electrophysiology talks. Really, my main work over the last 15 years was electrophysiology, and I developed a bunch of multifunctional electrodes, including, maybe you've heard of neuropixel probes that are now being used even in humans here at Harvard. I sort of was very involved in developing all of these probes, and also developing miniature robotic devices that you see the movie of on the right. These are 80 micron diameter glass patch pipettes that robotically go into the brain. So if you're interested in electrophysiology, multifunctional electrodes, and robotic microdrives for brain science, please let me know. I'm happy to talk about that. But this is going to be a talk about labels, imaging labels, and this is really a story of what I went through in developing this. So I started in Janelia in 2008, and these are some of my colleagues at Janelia. This is Jennifer Lippico-Schwartz on the left, Harold Hess, who hired me at Janelia, and Eric Betzig. And at that time, Janelia was sort of on the forefront of super resolution optical microscopy. And this is a technique that came about because Jennifer developed these photoactivatable GFP labels, and then Harold and Eric sort of got the light bulb in their heads saying, well, maybe this is a way for us to beat the long-standing optical resolution imaging limit. And so in the middle, what I show is if you have two emitting labels in optics, if they get too close, at some point you cannot resolve them anymore. However, if they're actually photo switchable and they're actually blinking, and they're not on at the same time, you can actually individually localize them and resolve them to better resolution that's typically possible by diffraction limits. And so you can, as opposed to getting images like this, which are standard in optical microscopy, they were able to push that optical resolution to something like this. And Eric Betzig was in 2014 awarded the Chemistry Nobel Prize for this work. So to me, coming in, I'm not an optics person, but I had a background in magnetic materials. I was always kind of curious, well, can we do something like that in MRI? Could we have a switchable magnetic label as opposed to an optical label in MRI? And I was very fortunate and honored to meet Dr. Alan Koretsky at the NIH, who was sort of the magnetic particle imaging guy at the NIH. And he was able to show that he can see individual micron scale particles in MRI, okay? And so what they look like this dephased parts of the image that look dark. But the issue is that these particles this imaging label is always on, okay? It's always on, and so you can't really make it blink. And why is that? So let's look at a little bit of the fundamental properties of magnetic materials that you learn about in school. So if you look at common properties of, let's say, pure iron or iron oxide, and I will be putting a red arrow basically at body temperature throughout this talk. Basically the curve of magnetic moment versus temperature is pretty flat for most magnetic materials. And so therefore you can't really change it magnetically in a regular setting. And then especially in typical MRI fields, these are moments versus field, the materials would typically in Tesla scale fields saturate. If you put iron in an MRI, it's going to be saturated magnetically. And so you can't really change that magnet inside the scanner in really any way. And so I thought, okay, well, that's the end of that idea of trying to make them switchable until I discovered this paper, okay? This is a paper from the 60s, early 60s. I want to note that this is before MRI was invented. Okay, so it's not like I invented some material. I just discovered this paper. And what's interesting about this, and I simply couldn't believe my eyes and I couldn't really believe my luck, okay? So what this data is, it's a data of magnetic moment versus temperature for a material. And notice that it's basically non-magnetic, 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 but around body temperature, okay, it goes through this extremely sharp, what is called a first order phase transition, from a non-magnetic to a fully magnetic material, okay? And additionally, notice that this is a data taken at half a Tesla, okay? So we have this material that in half a Tesla and around body temperature can be switched with slight changes in temperature from off to on, okay? So that was kind of my light bulb in my head that happened, and I basically got very excited, and just to show you that we can also switch these things with field. Here's another paper before MRI was invented in the 70s. So this is field from 0 to some value versus moment. And this is a data about 20 degrees below body temperature. This is a data about 20 degrees above body temperature. So in between is where the body temperature would be. Notice this moment goes paramagnetic, and then it switches on to something ferromagnetic. And then when you reduce the field, it switches back off. But these values of the field, this is 10 Tesla, okay? So again, body temperature, Tesla scale fields were switching these magnetic materials. So, I basically took these papers straight to Alan Koretsky, and I said, Alan, this is pretty interesting. Maybe we should put these materials into the scanner and see if we see something. And he got excited too, and he basically said, if you prove to me that you have these materials, I'll promise you we'll put it in the scanner. So I basically went to my boss and I said, yeah, I've been looking for these materials for 10 years. Would you fund me to kind of explore in this? And luckily enough, Tim Harris, my boss at the time at HHMI, said, go ahead. And I said, look, my family lives in San Diego. My buddies from grad school at UC San Diego are now senior executives at Quantum Design, which is a magnetometer company. you know, if you let me fly there, I have a place to stay, the measurements will be free, and we'll see if we can do this. So anyways, I flew to San Diego a few times with my samples that I prepared based on the recipes from this paper from the 60s. You know, long story short, eventually, here's the data that I presented to Alan. So this is a magnetic moment versus temperature at different fields, constant fields. Okay, so the green curve is 1 Tesla, the red curve is 3 Tesla, the blue curve is 5 Tesla, and the purple curve is 7 Tesla. And again, to this day, I'm sort of amazed that this thing just going boom to on and boom to off within 10 degrees C. And this is the body temperature here. So the 5 Tesla curve is the best match, okay? And so same sample, but now we're keeping the temperature constant and sweeping the field. 10 degrees below body temperature, red curve, green curve, body temperature, blue curve, 10 degrees above body temperature. Notice again, around 4 or 5 Tesla, this thing is switchable, okay? And the hysteresis is about 1 Tesla here. So luckily they had a 4.7 Tesla scanner, which is the closest I could match to the data that I took. This is my dear colleagues, Steve Dodd and Doug Morris, who were happy to help me and open their lab to me. So I would sneak out of Genelia and go do this. It's kind of my passion project. Here's their 4.7 Tesla scanner, and I get a kick out. And when we first started, we would write these, you know, time and temperature of the sample. We would measure, and but basically this is the punchline. We had a sample in a regular kind of phantom water or agarose with water tubing, to which we swept the temperature, and we had a MRI compatible optical fiber probe. And here's the image. This is the image actually of the fiber sensor. And here's the sample being imaged in MRI as we are sweeping the temperature up and down. This is what happens. So, you know, this thing really does magnetically explode essentially, and you can turn it on and off. And so what's sort of, again, to this day, shocking to me and very pleasing is that this happens at body temperature and 4.7 Tesla. Okay, so we have this magnetic material that I can turn from essentially non-magnetic to almost iron level magnetic at body temperature and Tesla scale fields. So this motivated us to push further, and just like in optical microscopy and fluorescence microscopy that I mentioned before, point quantum dots, for example, you have a lot of different choices in different imaging labels at different wavelengths and whatnot. And I want to say here that the same is actually the case for these materials. So here's the same material. By the way, I don't know if I mentioned, this is iron and rhodium. Only 2 elements, iron and rhodium, okay? And that's the material that exhibits this property. And basically the standard material science techniques of doping, like you would do in semiconductors, you tune the properties very, very precisely. So here's the data that I showed before on pure iron rhodium. You dope it a little bit with platinum or iridium or palladium, you shift these curves very nicely along these curves. And again, if I change the field, I sweep these curves to the left or to the right, depending on the field I apply. So again, this is the data taken. In this case, look at this, 0.7 Tesla. Okay. So there's a huge variety of things you can do with them. And so I happen to have 2 samples of iron rhodium. One was more pure than the other. And so, for example, the lower curve is for a less pure iron rhodium. These are the curves, switching curves in the field. The more pure iron rhodium is shown on the top. And so we put both of these samples in the same phantom, okay? And what do we expect to see? So at 1 Tesla here, which is going to be this image, what do we expect? We expect this one to be switching, but this material should not be switching at 1 Tesla, nothing going on. So when we did that in 1 Tesla MRI that they had at the NIH, this is what happens. Notice that the top sample does not change in contrast, whereas the bottom sample changes contrast very nicely. If we put the same phantom in the 4.7 Tesla, what do we expect? Well, we expect this sample to be saturated, and it is, and then the other one to be switching, and it does. So here we go. So this is a 4.7 Tesla. The bottom one is not changing, but the top is now being switched on and off. And it's not just iron rhodium. It turns out that this is a large class of material. Typically go under the name of magnetocaloric materials. They're very popular in magnetic refrigeration community and data storage and spin electronics. And so there's a lot of materials that I then started searching through. And the mission for me was always this: is this material switchable? Is this transition sharp? Does it happen at body temperature? And does it happen at Tesla scale fields? So all these things need to be satisfied for me to consider that material to put into the scanner. Okay, so is the transition sharp? Does it happen at body temperature? And does it happen in Tesla scale fields? So now I'm going to answer some questions, looks like. Switch. Yeah. Right, right. So the question was, what's the physical basis of this sharp transition, first order phase transition? It turns out that these are called magnetostructural transitions. So what really happens is there's a slight structural transition that happens with temperature. And we know that the exchange interaction is highly dependent on distance. And so it seems to be that the slight structural change causes the change in exchange interaction to flip this from antiparamagnetic to paramagnetic state and vice versa. So that's the fundamental reason. And I think your question is also posing, the next step, my next slide is that what I showed so far is this thing is non-magnetic and then turns into strongly magnetic. But this set of materials that I show here really have a wide variety of properties. So here's another material that I found where it has similar curve to iron rhodium, sharp transition around body temperature. This is 2 Tesla, okay? But most magnetocaloric materials actually have this opposite slope. They go from magnetic and then a sharp transition to non-magnetic. Again, this is body temperature here, and this is in 1 Tesla. I like this data because it also shows gadolinium, which has sort of this second order phase transition. It's a much shallower slope, and that's why we typically don't use it in this work. But so there's a lot of materials, and they actually have these transitions that can be positive slope or negative slope. And so this opens up, again, a lot of avenues, hopefully, and I'll show you some examples. So I started looking, for these other materials. I tried to find sort of commercial sources and only found one, and I'll tell you about it. But for example, here's the material that is in this category of lanthanum iron silicon materials. When it was first discovered, which is this black curve here, it has a sharp slope. It happens in 2 Tesla, but notice that the temperature is very, very cold. But what they discovered in Japan is that when they infused this material with hydrogen, they could very controllably, again, material science, shift this curve up and up and up to the body temperature, okay? And so, the slope stays sharp, the field stays large, but the sample is now at the body temperature. So basically, again, this company, Vacuum Schmelze in Germany, they're actually pretty big company. This is not a big part of their business. But I begged them basically for samples. They were kind enough to send it to me, but I was really going for particles because, we demonstrated that the physics worked, but I wanted to now push this to smaller and smaller particles. And a dear colleague of mine, Hatem Bidway from US Naval Academy, he then got a magnetometer in Annapolis. I didn't have to fly to San Diego anymore. I would just drive to Annapolis from Northern Virginia. But here are the curves. The green curve is a 3 Tesla, blue curve 2 Tesla, red curve 1 Tesla. Okay, so notice that this is a very different looking curve than iron rhodium. So different physics happening here. But as far as I'm concerned, the slope is sharp, It happens in Tesla scale fields, and it happens around body temperature. And I have these particles now. And so they're about 100 to 250 micron particles. What I did, I with my tweezers, picked the smallest particles of the sample that I got, placed them on a glass slide in an array, okay? And notice now this should be on when we're cold, and then when we raise the temperature, they should go off. And this is MRI at 1 Tesla. There we go. This again. So these things are switchable magnetically in an MRI around body temperature. Okay. So that was pretty interesting. Here's another interesting thing that I discovered when I overlaid the data for this lanthanum iron silicon and one of my samples of iron rhodium. Okay. This is at the same fields: 1 Tesla, 2 Tesla, and 3 Tesla. At 1 Tesla, which are the red curves, they sort of matched where the transition happened. One is positive slope, one is negative slope. So we put them in the same phantom, okay? And what do we expect? Well, if we're sitting here, this one should be on, this one should be off, and when I raise and lower the temperature, they should be switching in opposite sense. Okay, here's what happened. Now, it's not surprising that this works, but it's gratifying to see it working, kind of in front of your eyes in the MRI images. So again, my point is that these materials are quite versatile. They have a bunch of sort of these unique properties that could be quite interesting for MRI applications, because they are basically tunable magnetic materials in Tesla scale fields. So there's another question.
Sorry, this might be a silly question, but you're talking about body temperature, and I understand why that should be the case, but do you have to like measure like kind of like a local temperature, say if you were using these as materials that go sit once in the brain or something? Would that be, because that might be different from like the
I mean, so we do not believe, and frankly, I don't get much traction when I suggest that this could be a temperature sensor. Okay. So we're not proposing this as a temperature sensor. In this case, we're heating and cooling the entire bio. Okay, and we put our temperature sensor right next to the particles, so we sort of know what the temperature is around the particles that we're imaging. So does that answer your question, or?
Yeah, I just wanted to know, because then I know I'm not an MRI person, but they talk about all these. What is it, SAR, and then the temperature is changing during.
I'll address that. I'll address that in a little bit. So another thing that we did. So in everything I showed so far, we're heating and cooling the entire phantom, and that you can question that for eventual clinical applications. But so one thing. So how else can we heat these particles? Okay. Well, you can say you can heat them with lasers. We can heat them with RF. You can heat them maybe with ultrasound. And so I'm like, okay, how about ultrasound? And so what I did a little bit of searching, like who does ultrasound in the area where I live? Like who does maybe ultrasound and MRI? And it turns out there's a focused ultrasound foundation down in Charlottesville, Virginia. That's about a 2-hour drive south for me. And so I paid them a visit. They introduced me to a bunch of faculty members to see if anybody would be interested in collaborating. And my last stop on my visit to University of Virginia was Professor Wilson Miller, who does MRI compatible focused ultrasound. And so I showed him our topic and showed some of the same videos I showed you. And I said, this is pretty interesting, but it'd be nice if we can demonstrate that we can heat it with something other than the whole phantom. So he was happy to collaborate, and so Steve Dodd and myself, we actually drove down to Charlottesville on a Sunday, one day, and here's his focused ultrasound machine that's MRI compatible. This is a 1.5 Tesla scanner, and again, I put individual particles in his sample holder, and what you'll see here, I don't know if you can see, you can probably see the outline of the ultrasound actuator that focuses ultrasound energy to a point. So we're going to first focus the ultrasound on this particle, and then you'll see this actuator switch to the left and then switch the other particle. And the blue arrow is sort of the direction of the polarizing field, if you will. So here we go. So first, then you see the actuator shift to the left. And then I'll play this again because I think it's fun. And we spent the whole day down there, and eventually sort of it goes so well. Then he was sort of driving this focused ultrasound beam. This is now looking at the surface where I had 4 particles, and we're sort of driving around and switching these particles with sequentially with focused ultrasound. Again, it's not surprising this thing works, but it's gratifying to see it work in a 1.5 Tesla scanner. Okay, so the big question remains: can you go to cell size dimensions? Okay, because everything I've shown you so far is 100 micron size or larger, millimeter scale samples. And the question was always coming at me like, okay, but does this still work when you push it to cellular dimensions? And so again, I go on my search and who would do this, and it turns out not that many research groups around the world do iron rhodium deposition, but I found this poster, and it was actually not a published poster anywhere. It was simply a poster on the internet by this group in Brno, Czech Republic. And so I look at this poster. It has iron rhodium. It has particles that are submicron in an array. And again, I find this simply amazing. You see this, he images them in magnetic force microscopy in 0.3 Tesla. And it's just sort of amazing to me. They're magnetic, but then as the thing cools, it says as if these particles disappear, like poof, they're gone. But they're not really gone, they're magnetically gone. They're still there. And so they simply go from magnetic to non-magnetic. Okay? And so I reached out to Wojtek Uller at Brno University of Technology that published this poster on the internet. And this, again, describing the same story and what we're doing and what we're interested in. And he was happy to collaborate. And so we launched a collaboration with him. And we said, we want the sample that's, for MRI kind of thing. And so we send him this diagram, top left. He sent us the sample that sort of matched that bottom left. We wanted to have kind of a calibration, larger area, but then we also wanted to push these down to cellular dimensions, 10 microns, 5 microns, 4 micron diameter particles of this iron rhodium. And here's the sample in MRI. So again, these are not particles that are cellular dimensions, okay? Biological cell size. And first we did a temperature sweep. And again, you can actually see some of these 10 micron diameter particles. They're only 200 nanometer thick, actually. So these are very thin films. So definitely volume of a biological cell. And the exciting thing is that we can see them and we can still switch them. They're fine. They work just fine in submicron dimensions. And so the final really question was, well, you're switching all of these materials so far with temperature. And that's perhaps problematic for clinical studies in the future. And so, but there's 2 ways to switch these materials, as I've shown you in the beginning. These curves look very similar, but actually different. The one on the left shows a constant field, and I sweep the temperature. The one on the right shows a constant temperature, and I sweep the field. Okay, and notice again the switch happens around 5 Tesla. So if we're sitting here at 4.7 Tesla, okay, and if I were to add within the scanner temporarily a field of, let's say, 1 Tesla, I should be able to drive this curve to this point. And then if I'm able to subtract the field in situ by, let's say, 1 Tesla, I should be able to drive this particle back down to off, right? So if I, within the scanner, am able to add plus or minus 1 Tesla, I should be able to be switching these particles on and off, okay? So the first suggestion is like we should try to find an MRI compatible TMS coil, something like that. So we searched for that at NIH, and that probably would have been a good route, but somehow it didn't happen. Alan was just happy to invest in a field shifter, but just to kind of show you that in principle this should work, Steve Dodd took these images at 4.7 Tesla of the sample from the Czech Republic, from Brno. And this is where the particles presumably are off, right? And you kind of barely see them. And if you image that in 11.7 Tesla, this is when they're on. And you can actually see these particles very easily, including down to 5, 4, 3 microns. Of course, you can see a much higher signal to noise at 11.7 than you would at 4.7. But in principle, they should be switchable with field as well. Okay. So Alan very generously agreed to invest in a field shifter from a company called Resonance Research here in Boston. And then at that point, Natalia Gudino joined the effort. She was interested, and she's sort of the RF expert at the NIH. And so I was very happy that she was happy to help. And so she worked on the electronics to drive this shifter that's inserted into the 4.7. And the beauty is that it applies this extra 1 Tesla added or subtracted from the main field, but it doesn't generate any external field, so it doesn't quench the system. So, so here's Steve, here's Natalia with their gear feeding this field shifter. I do want to point out this whiteboard, which we haven't erased for 8 years. Okay? So it's still there, So, but then we tried. So here's the experiment. So we have the sample, and it's sitting at 4.7 Tesla. And what we do, we image at 4.7. Then we briefly apply a pulse of, in this case, 0.8 Tesla, and presumably would have turned the particle on, right? And then we image, and then we apply a negative field of 0.8 Tesla. So we subtract from the main field, and presumably we will turn the particle off, and then we image, and then we repeat that multiple times. So now we're not switching the particle with temperature, we're switching it with field, okay? So the switching time of these metamagnetic transition is literally unbelievably fast, like picosecond. That's the word. So way shorter than anything we do in the MRI. So it's at least nanoseconds, but probably faster. So this transition is extremely fast. The only thing I can possibly see that could maybe delay it is some kind of eddy current effects or something like that. So we don't image during this application of the shifter field. So we image at 4.7. We briefly add a 0.8 Tesla field, just up and down. Image at 4.7. Correct. So let's go through the curve right here. Here's the curve that I showed earlier. Okay, here is 4, 5, 6 Tesla. We're sitting at 4.7. So presumably we start here on this hysteresis curve. When we go up in field up to 5.7, we drive the curve someplace, and presumably when we turn that off, the particle is now here. Okay, it's stuck here, it's latched. And then we image, and then we again apply a field, but now in the opposite direction, and then we drive this particle down this curve to here, and we presumably should be here. And basically, so we are imaging between this state here and this state here. Oh no, it stays there forever. It's not. It only will go out if we take the sample out of the field. But that's basically like we do, taking this curve down to 0. Oh yeah. If that's what you're asking, it's permanently latched there forever, let's say.
Long enough.
Long enough for sure, yeah. Yeah, that's exactly what this is. So hopefully everybody kind of understands the experiment. We're imaging at 4.7, and we briefly apply positive and negative field to switch this particle with field in this case, not temperature. Temperature is constant. So here's these images that we obtain in sequence here. Hopefully you'll see this particle here, they're switching. I'll play this again. So to me, what's exciting is that these particles are small now. They're down to biological cell volume. And so this is really, my presentation, my proposal and demonstration to you. That there's this large class of materials, which typically go under the name of magnetocaloric materials. Hopefully I've convinced you that they're switchable in Tesla scale fields and at physiological body temperatures. Hopefully I've convinced you that we can switch them both with temperature or magnetic field. When it comes to temperature, we can do it with focused ultrasound or heating the whole phantom. I think these materials are quite interesting because they come in a variety of sort of forms, and they can have a positive and negative slope of switching. They can switch at different magnetic fields, as I've shown you. And what's exciting is that they're switchable now down to cellular dimensions. And when they're that small, they're still visible to us in an MRI. And so we feel like we basically have done all the physics demos that we need to do. And we're now basically ramping up collaborations with Brno in Czech Republic and others to try to get them basically detached from the surface, functionalized so we can attach them to cells, place them on cells and see how that goes, and then track the cells with these things. But we think this thing is real, it works well, and with effort we'll be able to basically label single cells and track them for a long time with these blinkable labels. So I think with that, I'm done. I want to thank a lot of people. First of all, Steve and Natalia and Doug and Alan, who just opened their arms to me when I first came with this idea into their lab. Tim Harris, who was my boss at HMI, who let me pursue this topic. Bruce and Bill were the machinists that machined these samples for me when we first started. Neil and Stefano at Quantum Design will help me measure. Barbara and Alan at the Naval Research Lab, who helped me prepare the samples. Hatem and Virgil were kind of partners in discussing magnetocaloric materials. Professor Wilson Miller and Katja Gilbo at University of Virginia with their focused ultrasound. Wojtek at the Brno University of Technology in Czech Republic. Vacuum Schmelze, the company that let me have some of their powder samples. And Mom and Dad, of course, for hosting me in San Diego during my research trip as I was chasing this topic. So with that, I am done and happy to answer any further questions from the audience or Zoom. Thank you.